presentation mode. So let me just do that. So this is the, so I think it might be the third or fourth webinar that we've done just for the members only. So this is, this webinar is really just for you guys who are kind of members of the club. So I'll just give it another sort of 30 seconds. So I can see that I think it's gone on to YouTube Live now. So we're actually streaming. So today we're going to do a webinar focused on one of our members called Abril. So I'll just say hello to everyone. See, let me just put into live streaming. Hello, guys. All right, cool. All right. So what I'll do now is I'll do a. Today we're actually going to focus on Abril. I've known Abril for possibly at least, I feel like 2 years. We met in Malaysia at a Biosensors Congress, and I know he's really good friends with Hasim and their colleagues. Now, Abril is quite interested in, when I first met him, I think in a part of our conversation, we found out that actually Abril's, he did some of his studies and some of his academic work in London. I think it was at something like the School of Infectious Diseases. So he's quite interested in parasites and sort of detecting parasites and correctly identifying parasites. So today's webinar is a little bit more, let's say, different because Zim and Peacock, we like to make biosensors, and we talk about the detection of glucose, and we talk about the detection of lactate, and we talk about the detection of proteins. But today, we're actually going to talk about the detection of a parasite, which I find really interesting because, the parasite that we're talking about in today's webinar is 20 micrometers in length. So if you think about, most of the time, we're trying to detect something that's, some nanoamps or some Armstrong's size, and then now we're talking about, micrometers. You can see the different scales that sensing has to take place on. So what I'll do is I'm just going to give a quick introduction to the particular topic that we're going to cover today, which is actually something called, so the parasite of interest is actually called Trypanosoma evansi. Now, Trypanosoma evansi in Indonesia leads to a disease called Surra. So I hope my pronunciations are good on this. So it's a nasty blood, well, I think it gets into the blood, and it leads to a sort of chronic disease. And here you can see that unfortunately, the horse is very kind of wasted. So pretty unpleasant. And I think it's, there's actually, at least these trypanosoma type parasites, they're very global, and I mean, they're sort of touching just about every continent in the kind of equatorial regions. So you've got this parasite. Unfortunately, it gets into the livestock, and you can sort of, it causes a disease called Surra, and if you Google that, you're going to find a lot of pictures of, unfortunately, emaciated livestock. Now, Surra can be detected by several methods, and this is a really interesting thing. That at Zimmer Peacock, we are electrochemists, so we like to make biosensors, and I have an example of a biosensor here. This is a glucose strip. It's got a little capillary on it, and that capillary sucks in blood. There's some enzymes in there, and it can detect the glucose in blood. So we like to make, and the method for getting the signal out of that sensor is electrochemistry. So we like to make biosensors. Now, the question is electrochemistry always the best technique in order to, I just want to check something. Is electrochemistry always the best technique for detecting the analyte of interest? And in this case, I think electrochemistry can help in the detection of trypanosoma, and in the sort of identification of Surra's disease. But it's not the only technique. So let's have a look at the techniques that that have been done. So one of the techniques is blood film. So this is a very kind of conventional technique. And what you do is you put a drop of blood, you make a thin film of that drop of blood, and then you literally look under a microscope. So, what's interesting about that is, if you want to detect this parasite, then looking for it under a microscope is not a bad technique. Now, when I was researching for this webinar, and the things that April was interested in, I was then interested in the fact that actually there was also a technique, I think, that's related to the blood film technique, which was hematocrit centrifugation. So basically, you take some blood, you spin it very fast in the centrifuge, the blood separates out into its components, and between the plasma and the erythrocytes, so these, in a centrifuge, the components in blood are separating due to the, I want to say centripetal or centrifugal force, but you're rotating very quickly, material moves through the tube depending on its mass, and the things separate out. But what's really interesting is that the trypanosoma actually ends up being concentrated in a thin, they call it the Buffy coat, but it seems to be a sort of thin layer between the plasma and erythrocytes, and it gets concentrated into there. So that I would consider an improvement over the blood film technique, because with blood film, the parasite could be anywhere in the sample, but when you actually centrifuge it, it ends up concentrating the parasite into a much smaller volume. So that means if you're trying to positively spot and identify the parasite, obviously that's an improvement because you've concentrated the sample. So I like those two techniques, and one of them is quite simple to perform, the blood film, and the other one you need a centrifuge. Now, I've got here something called complement fixation tests. Now that's actually from April's paper, so he already knows about this technique, but effectively you cause a kind of a clumping of the reagents if the person has had the trypanosoma. So it's almost like a sort of colorimetric test or a clotting or coagulation of particles, and it only takes place when that person's unfortunately had the parasite and therefore the disease, and therefore probably has antibodies that reflect the fact that they've had the disease. But it's quite a clear signal, and effectively what they have is a little card, and they view it, and if they get number 10, where it's sort of it's a kind of there's transparent regions and then more opaque regions and transparent regions, then I believe that that's the positive. Whilst researching this, I also found that there was also a DNA papers out there. There's also antibody tests as well. So we've got two kind of microscopy type tests, and then 3 biochemistry type tests that we're going to discuss today. So I'm going to talk about the microscopy tests to start with. And the blood film, I actually think this is a fairly easy test. But the thing that makes it, you do need training. You need a spot. Well, first of all, you need some microscope slides and a cover plate. You need to be able to put a spot of blood onto that. Then you need to scrape it thin, and then you need to drop a cover slide over the microscope slide, and I also think that there's some staining solution involved. And then you have to look at it under a microscope at 1,000 times magnification. So it's fairly easy, but you do need some equipment and some reagents. And then the trained expert will be able to spot the parasite. Now, the purists out there will say, well, how do you know it's the particular parasite of interest? And when you spot that parasite, whether you then have to go on to do speciation really depends on your application. And I would argue this, that biosensors and low-cost testing, these are really kind of screening tools. And if you want a really accurate answer, then the sample of blood has to go back to a central lab where, a real sort of, I want to say, pathologist or parasitologist looks at it and says, right, yeah, I've really identified this. But what are we trying to achieve sometimes in biosensing is, you need to do field tests. You need to know whether this guy's got a problem or not. And if he's got a problem, then you have to go on and, dig in deeper. But so I think that it's actually okay sometimes to just spot the fact that there's parasites there. Now, what parasite it particularly is, it's nice to be able to do that also in the field. But it doesn't have to be the first product or the first thing you do. It could be enough to be just a screening tool, an in-field screening tool. If people test positive, then the sample has to go into the lab. If you don't have an in-field screening test, then everything has to go into the lab and has to go in front of sort of PhD-trained scientists. So now we touched upon it a couple of slides up, but the other way of doing this is to take the blood and put it into a centrifuge and actually separate all the blood constituents, and then the parasite will actually get concentrated in the buffy layer. And then you have to kind of look at that buffy layer and say, can I see the parasites? And then you can count them, and and if you count them, then you can sort of also quantify, how serious is this infection. Now, when I was reading all of this, especially when I was looking at the idea of creating a film of glass, I'm just going to click this link actually, because what I was thinking when I when I was preparing these slides, I was thinking, well, what's the easiest way of preparing a thin sample of blood? And I mean, if on our website, I've played these videos before, but we do this quite a bit in biosensing. So at Zimmer and Peacock, we do a lot of disposable medical diagnostic development. And here I'm showing you now we are trying to create a thin or a sample, and what we're doing is sucking into a capillary. There we go. Now, I also realized that if you did that with blood, you're actually creating basically a thin layer of blood. So let me show you why I'm saying all of this. So I'll just bring that back across. The reason I'm saying this is because at the moment, one of the ways of detecting the parasite and therefore kind of diagnosing the disease correctly is to take some blood, and then the blood has to be prepared by somebody who's semi-expert on glass slides, and then it has to be looked under a microscope. Well, in fact, if you want to create a thin film of blood, you can just do it automatically if you use microfluidics and capillary fill. And I suppose, to the members, if you guys are not sure how to do this kind of thing, then our website is a good source for this kind of information. And also, you are members, and if you're a ZP Developer Zone member, and this webinar is for you guys, then we have the Discord server, and we all sort of could chat down there and discuss this. But what I'm saying is, all right, so in this method for trying to identify the parasite, you have to very carefully prepare a thin layer of blood. It's actually easier to have a capillary fill device like this. And to make the point, I did have a go at a quick experiment yesterday afternoon. So I took a glucose strip that I had in the office. Clearly, I'm going to have these kind of things because what do we do day in, day out? We develop biosensors, we test biosensors. So I took a glucose strip, I pricked my finger, I got a drop of blood, I wicked my drop of blood into the microfluidic chamber of the glucose strip. In fact, it was this one. And so I did all that process yesterday. And then I looked at it using a really quite a low-cost microscope. I'll just grab the microscope, in fact. So I had one of these microscopes, and I noticed actually that this microscope had a maximum magnification of 800x. And I think to really do this well, you probably need 1000x. But I would say this, that, you know, if you're trying to do a in-the-field test, it doesn't always have to be perfect. Hi, I can see the guys here at the moment. I'll just say hi to Hasim. Yeah, cool. So I did a simple test yesterday afternoon where I'm trying to, you know, say, okay, one way of identifying this parasite and therefore sort of diagnosing the disease is to form a thin film. In fact, microfluidics and capillary fill sensors is a good way of doing that. And then taking a low-cost microscope. So I did this in my office, as I say, this particular microscope. I then sort of did a quick experiment. So the idea is this. This is the zoom in of this sensor. So you can see how much I could actually have magnified that. And I imagined that I was actually trying to spot a parasite that was potentially 24 microns long. I'm not suggesting it's easy to do that. But what I am suggesting is this. I think even a low-cost microscope has the ability to potentially do that. And, you know, I even zoomed in then electronically. So you can get quite a good magnification on these kind of devices. And so I am an electrochemist, I am a biosensor developer, but I'm also a pragmatist. And at ZP, I think all of us in the developer zone, we have to be pragmatists. If there's different ways of detecting things, then we should promote it and discuss it. So for this one, I do think, you know, you can use some of the principles from biosensing, like microfluidics. But in fact, then, let's think that the biosensor could actually be the camera or the microscope. So I've put a picture here of I've had one of these microscopes myself in my time. It screws on the front of a smartphone. So now we have the ability to create a film of blood quite readily. I did that in my office yesterday afternoon. Whoops. So we can create the film of blood quite readily. And then, if we can position the capillary underneath a microscope, particularly one that's connected to a smartphone, we suddenly have the ability to capture the data. Yeah, okay, I can just see that April's here as well. That's cool. So we have the ability to capture the data, and now we're on our way to really making a field application, because what we can do is we can transmit the data you know, into the cloud. You know, you can literally just send an email if you so wished. You can send it as an attachment. And obviously at Zimmer and Peacock, we're much more interested in making it a product. You know, so we would be sending the data to a cloud server. And in fact, in the first instance, if I was trying to write a paper around this, the guy that I would have looking at the images would be April. He would look at it and almost score it and say, right, you know, I think I can see the parasite or not. Because then what he's able to do is he's able to then put images into buckets. He can say parasite present, and then in another folder or bucket, parasite not present. So what he's doing is he's building a training set of images where the parasite is present and images where the parasite is not present. And if he can do that, then he can find a colleague or collaborator who can do artificial intelligence. Because what he has is he has a training set of data. He has known positives and known negatives, and they're images. And artificial intelligence is really good at looking at images. You know, when people talk about image recognition, they're really talking about basically photographs and video. So at Zimmer and Peacock, yeah, we are really focused on biosensor development. And as part of that, there's microfluidics. And I'm also pragmatic and saying, actually, you can use some of the things we discuss, but the biosensor in this case wasn't electrochemical. The biosensor was the phone camera, and the phone camera has had. It's not ridiculous to say, you know, there's been billions of dollars spent on the development of smartphones, so they are really quite a good technology. I am gonna sort of dive into some electrochemistry as well here, but I just wanted to kind of share that with you guys as well. Now, I think there's a pretty good way of enriching the sample, and that's to do centrifugation. So we did touch upon this on at least 2 slides now, where, you know, in order to concentrate the parasite, if you centrifuge it, then the blood components separate based on their kind of their density or their mass, and actually ends up concentrating the parasites in quite a small zone. And if you can image that, then you'll have a greater chance of spotting the parasites. And the big concern with actually having to do centrifugation is you need a centrifuge, and centrifuges I have one in this office somewhere, but they're pretty big and bulky. And I just want to pick up on this paper that unfortunately I thought about this about 5 years ago, but I didn't do a paper on it. But these guys did, so we must give them credit. And what they've done is they've turned a fidget spinner, or the concept of a fidget spinner, one of these devices where you kind of spin it in your hands, and it's a little mini centrifuge when you think about it. So they literally have turned the concept of a fidget spinner into a centrifuge. So I think that this is interesting because now you can take your drop of blood and you can still do the capillary fill that I was talking about, where you can form a sort of thin layer of blood, but you could have also centrifuged it in the field and actually done some separation of the erythrocytes out of the sample, and that may have made it easier to then spot the parasite itself. And then the workflow is the same thing again. So you then can use the microscope on a smart camera or use one of these USB microscopes, get the image, send it to an expert, and the expert can start looking at the data and categorizing it as positive samples and negative samples. And it can then go into an artificial intelligence effort where you try to get the computer to be able to identify whether the sample has a parasite or not have a parasite. So I'm just sort of saying that, in fact, you know, some of the what we're really talking about here is lab on a chip. So lab on a chip, you know, is the idea of processes that we would normally do in a lab. We've reduced them down to microfluidics and lab on a chip. So effectively, this is a lab on a chip concept, that you put a drop of blood in there, it acts as a centrifuge, but it also acts as, it makes the blood, rather than being a drop of blood, it makes it a thin layer, so we can actually look at the blood under a microscope. And if that microscope happens to be on a smartphone, I really like it because those cameras are really good quality cameras these days. And once you take a picture on a smartphone, either because you've developed an app or you're just taking a picture and then emailing it across, suddenly the data can go into the cloud, it can go to an expert, it can, you know, and it becomes a sort of, at least a way of gathering data in the field. So my last point on this particular section of today's webinar is really about artificial intelligence. We are using AI quite a bit at Zimmer and Peacock. And most of the guys who are really good at it, you know, are basically young students. I mean, they are really good. And the reason being is that they're all programming in Python. I have a little play on Python as well. I mean, you know, you can download Python off the internet. It has AI modules on it. Those are also downloadable. So in fact, the barrier to getting into AI is unfortunately having a laptop or a computer or a Mac. Otherwise, the software is all free. So if you can gather these kind of images, then you can start using AI to train the AI algorithm to say, is there a parasite present or not? And we've been really startled by how good actually this kind of software is. So the idea would be, take a drop of blood in the field, you suspect there might be a parasite present, create a thin film of it, maybe even centrifuge it in the field if you use the fidget spinner concept. And then the image will go to the cloud, and you can run AI algorithms on it and try and train an algorithm to spot whether the parasite is present or not. Now, I'm going to talk about complement fixation test. So this is actually April's paper, so I have to cite it. But he has a way of, if somebody's had an infection and they've therefore got sort of the antibodies to that disease, they have a way of doing a biochemical reaction. And if the biochemical reaction, I think this is the positive sign here, this number 10 here, I know it is. That's a positive sign. Now I know, so effectively what happens is we have antibodies and antigens, and in the presence of some sort of sensitized, in this case it's red blood cells, they clot together, or they stick together, and that's what forms this kind of, you have transparent regions and opaque regions, and transparent regions and opaque regions. So the person's had the disease, they probably have some antibodies to it. You put in a sensitized material, and if it clots and forms these little these little clumps, then that's a positive sign that, that's a positive test. And I read through April's paper, and I can see that that there's some good work on it from the Institute of Tropical Medicine in Antwerp. I want to bring this now back to electrochemistry, because even though I'm very keen on the whole camera and smartphone device, at Zimmer Pre-Go, we are focused on electrochemical biosensors. But when I look at a test like the one that Abril did in his paper, at the moment, the problem is that somebody has to interpret that data. They have to read that. Now, you could either read it using a smartphone, so you could take a picture of it, send it to somebody like April, and he could actually identify, the outcome of the test. But also, you could actually perform that test on an electrode system. So what I'm suggesting here is, we did a whole talk last week, 60 minutes, on impedance spectroscopy. And I think impedance spectroscopy also has a role to play here. Because effectively, you have two electrodes, and the resistance between those two electrodes will be different depending on whether you're in this M1 situation or in this positive situation down here. And the reason being is this. When we were talking about impedance spectroscopy last week, we said electrodes can be modeled by a circuit where we have RS, which is the resistance of the solution, double layer capacitance, that's capacitance at the working electrode, charge transfer at the working electrode, and then diffusion to the working electrode. So when he has this kind of clotting going on, RS is going to reflect it. The RS value here is going to be different from this M1, M2, M3, M4, M5. RS is going to be different each time. So that is a way of doing the test, but actually in analyzing it by an electrochemical method. And I just want to put this up as a reminder from last week's webinar. But I'm basically saying what you can do is get one of these, or get yourself a conductance sensor and repeat the test on that conductance sensor, or do a very similar test, and investigate by impedance spectroscopy. And I strongly suspect that if you do a frequency sweep, you are going to find an equivalent circuit somewhere, and probably the RS value, or one of these circuit elements, will be very sensitive to the to whether the test is a positive or negative. And what I find interesting is I did find a paper that actually, was kind of in agreement with my idea. So I had the idea first, and then I went looking through the literature. And there's never an original idea. Somebody else has always thought of it before. So this paper was similar. It wasn't identical, because here they were using latex beads, but they basically had these kind of antibodies on the latex beads. And when the antigen came along, it forced the latex beads to agglutinate, they called it in this particular paper. But what was interesting is actually the fields, when you're doing the impedance spectroscopy, the fields actually brought the sort of these particles all between the two electrodes, so it actually kind of enriched the sample. So I do think that some of the work that Abril's done in the past could actually be translated onto an electrochemical sensor, and I think impedance spectroscopy could be quite an interesting technique to do that. I also, we've talked every week. We probably mentioned some sort of ELISA assay at some point. So obviously there are ELISA assays existing for the Trypanosoma evansi, and if an ELISA assay exists, then you can translate ELISA assays fairly readily onto electrochemical sensors. So now the thing about ELISA assays, people start to like things like ELISA because it's more specific. So earlier on I was talking about you can use you potentially could use microfluidics to prepare a blood sample. You could use a smartphone or a USB microscope to gather the data in the field. The problem with some of that is it could have been another parasite, but depending on how depending on if it's enough just to identify that they actually have a parasite, then what I described there is good enough. If the test has to be more positive, or more precise in terms of it's not just a parasite, but it's a particular parasite, then you probably have to start getting into some of these biochemical tests. So the ELISA assay does exist, and if it does exist, then it can be translated onto an electrochemical biosensor. And we have talked about this quite a bit. And I actually, I found a particular paper. I couldn't find one for your particular parasite, Abril, but I did find one on Chagas disease, or Chagas disease. So I think this is quite a good, it'll give you some good insight into actually how to do this, because obviously the diseases are quite similar. So what you can learn from this paper can also be translated onto an electrochemical paper. And I thought it was very interesting. There's nothing new in the world, because when we were talking about Isa's work, we were talking about gold nanoparticles on carbon electrodes, and it's come back again. So I do like the fact that there's a consistency, that what we talk about, it's not just our opinion. Yeah, these techniques are used quite heavily out there. Finally, DNA testing. I have reached out to the authors of this paper to try and get a copy of it. They're on ResearchGate, so if you're looking to get a copy of this paper, I have reached out to the authors. So, and I bring it up because, in fact, when we were doing a talk for Sarif a couple of weeks ago, there was a question about, well, who's going to do the DNA part of all of this? And I said, well, actually, at Zimmer Peacock, we're quite interested, and we have actually started up a sort of molecular biology group just recently. But I also noticed that, I'm sorry to tell everyone, April, but you've also got quite some good DNA and some good genetic techniques. So I think this paper is interesting because it shows that there's a DNA way of, and DNA is kind of one of the gold standards. If you really want to do a speciation on something, you want to say absolutely, that is the species that I think it is. And DNA is the good way of doing it. And at Zimmer and Peacock, we kind of say that if there's a sequence of DNA that's indicative of that parasite, and you know what that is, and I think this paper is a good place to start, then you can have what's called a capture strand of DNA. It binds with the DNA that you're trying to identify, and effectively they form a double-stranded DNA, and that changes the electrode surface. And then we can see that change in electrode surface using maybe impedance spectroscopy or cyclovoltammetry or square wave voltammetry. So I have brought the talk back to now to some of the things we've talked about in the past. You know, that I know at ZP, you know, we are famous for biosensors and electrochemical biosensors, but it doesn't mean that I wasn't interested in talking about the phone and the use of the phone also in doing parasites, at least parasites, if not identification, you know, parasite screening, let's say. So Hasim, I know that Hasim and Abril are online. Hasim dropped me a quick comment saying that you guys are there, so that's cool. So I just want to kind of do a little bit of a summary, which is, you know this already, Abril, but you know, parasites can be detected by visual systems. And if you guys are kind of thinking about research directions, I would definitely have a think about, you know, using the camera on a smartphone. You know, you can also, I mean, I like the cost point of these kind of microscopes. If you can get a good enough image of one of these microscopes, then that also helps with the kind of field or the field spotting of the parasites. And I like the fact that then the smartphones give you immediate connectivity back to either experts in the lab, or eventually you can teach algorithms to do the data analysis as well. And we can also do it by classical electrochemical techniques. So you can do this by the sort of agglutination technique that you've done in the past. Could probably do it by an Elisa assay. We can probably do it by a DNA detection. And I like the fact that, you know, gold electrodes can be used again. Gold nanoparticles and carbon can be used again. Electrochemical impedance spectroscopy, that can be used again. And it, you know, I mean, self-assembled monolayers. Every week we talk self-assembled monolayers. So I do like the fact that we are building now upon things that we've talked about, but now we're talking about their application. So I just check in the time. It's 8:38 here in London. I do appreciate the fact that you guys come every week and we do this kind of one-on-one webinar. You can see that I really do think about this quite a lot, you know, and I like it because I get to understand, you know, us all, you know, basically through our work. I want to say I'd be quite keen to do a members only workshop in Indonesia in 2021. So I'll maybe mention that now in the Discord server. And Hasim, if we can figure out, you know, when in 2020, because I would like to come to Asia this year, but I don't think it's going to happen because of, obviously, unfortunately, COVID-19. But let's start thinking about maybe having a workshop in Indonesia next year. And yeah, I'll be open to doing that. So I appreciate it today. If you've got any comments, I'll be on the Discord server in a bit. I'll also be dropping a link to this on YouTube, so you guys can find it there. And I'll also share the notes as well through the Discord server as well, so you guys can read that. So I hope that was useful. I'll just check, make sure I didn't get any messages from Hasim, just in case. So if I did, Hasim, I'm sorry, but no. Okay, yeah. Yeah, so Hasim says, put your questions here or we'll jump to the Discord server. So yeah, I think we're cool. All right, guys, so thank you again for this. Just lastly, the reason I put the picture of the FoodSense and the FoodSense app up there was this. If you guys are developing biosensor applications, I am interested because I'd like to eventually maybe put them on the FoodSense. And even if you're developing applications where we're using the smartphone and the camera, I'm also interested in that, because for me, food sense doesn't mind if it gets the data from an electrochemical sensor or whether it gets the data from the camera. In the end, it's all data. So I just want to kind of put out there that even if Haseem and Abra will end up using cameras and electrochemical sensors, they're all compatible with the things that we're interested in at ZP. All right, guys, I've said enough now. So yeah, like I say, watch the Discord server, and I'll put a link to this on there shortly. All right, cheers, Haseem. Thanks for getting everyone here, and I appreciate it. And have a good evening, guys.